Welcome everyone to our session on neural tissue engineering. I'm Chris Adams. I'm a senior lecturer in neuroscience at Keele University. And actually, I've put this session together with Divya Chari, but along with Stuart Jenkins, he's one of our speakers as well. We're part of the Neural Tissue Engineering at Keele Group, or NTEC for short. And the reason we wanted to put on this session is we really want to raise the profile of neural tissue engineering and what it can do, and maybe start setting up kind of local or international networks as well. But I'll talk a little bit more about that later. So how the session is going to run, I'll pass over to Divya, who's going to say a few words kind of introducing neural tissue engineering. We've got 4 speakers then, and then I'll kind of close the session with a few comments at the end. So I'll pass on to Divya. Thank you.
Thanks a lot, Chris. So firstly, I'd like to thank all of you for being here today. And I'd also like to thank the British Neuroscience Association for giving us the opportunity to showcase some of the recent research in neural tissue engineering. So for those of you who don't know, what is neural tissue engineering? Well, it's a relatively new sub-branch of the wider field of tissue engineering, and it focuses on repairing or replacing damaged neural tissue. And as a field, it integrates the use of biomaterials, scaffolds, cellular engineering, and drug and gene delivery technologies. So speaking from a personal perspective, I had a relatively conservative background training in neuroscience with a PhD in developmental neurobiology, where I studied the development of the mammalian visual cortex and how the multi-layered visual cortex was set up. And then that was followed by postdoctoral training and a fellowship examining the causes for failure of repair by oligodendrocyte precursor cells in diseases, demyelinating diseases such as multiple sclerosis, and the relative benefits of progenitor stroke stem cell transplantation therapies. And through my training, it became evident that neurobiological studies could provide really profound insights into developmental mechanisms and the causes for repair failure in the nervous system. However, it also became clear that the biological tools that were available were relatively inadequate in being able to develop effective interventions to address these situations. So when I moved to Keele to set up my own laboratory, I had the first opportunity to integrate into a large tissue engineering group, but they were primarily working on orthopedic and musculoskeletal applications, and they were very much the early adopters of these interfaces with engineering technologies. And it allowed me for the first time to integrate my research in neuroscience with physicists, chemists, and engineers, and that kickstarted an entirely new phase of my research career. And it also rapidly became evident to me that engineering tools could offer solutions to develop novel therapies and interventions for intractable neurological conditions. Now, for example, we know that the highly aligned radial glial scaffold plays a critical role in setting up the layered cortical structure, and that is disrupted in some neurodevelopmental conditions. But now, using aligned synthetic nanofiber scaffolds, we have an expert here in James Phillips. So using synthetic nanofiber scaffolds seeded with astrocytes, we can now create an artificial scaffold of radial astrocytes. Something like cell loss, for example, in transplantation had always been a major issue in transplantation studies, but we now have jelly-like polymer hydrogel technologies as protective matrices to encapsulate cells during the implantation process and protect them from immune attack. Hydrogels are also being used very effectively to develop neuromimetic models for advanced brain modeling and to simulate the stiffness of native brain tissue or native neural tissue, which plays a critical impact on cell fate. Graft cell tracking also, for example, has always been a major issue in cell transplantation, but chemists and chemical engineers can now synthesize highly versatile and multifunctional nanoplatforms, which can be functionalized in myriad ways, for example, to develop tools for genetic engineering, but also incorporating a metallic core that can be used for cell tracking and MRI applications using technologies widely available in hospitals. So we believe that the field of neural tissue engineering has a great deal to offer the fields of regenerative neurology and for basic neurobiology studies. And we've got a great lineup of speakers here. Thank you very much to them. And we look forward to hearing some of the cutting-edge research that they've been engaged with in this field. So without further delay, I'm going to hand over to Chris. Thanks, Chris.
Thanks, Divya. Yeah, thank you, Divya. It's a great intro. So I'm going to pass over to James Phillips. So James is a professor of regenerative medicine at UCL. But also co-director for the Centre for Nerve Engineering. And importantly for us, he's also Vice Dean in Innovation and Enterprise and has actually got experience in spinning out a company and that's something that I'll probably talk about a little bit later as well. Yeah, thanks, James.
Has someone got the clicker? Here it is. Brilliant. Thank you very much to Chris and to Divya and to the BNA for putting this on. It's really great to have the chance to come and talk to this meeting. A very long time ago, my original training was in more fundamental neuroscience, and since then I've kind of progressed into more engineering, but it's still always kind of cellular neuroscience for me. So as Chris mentioned, my lab's based at UCL, but I'm also co-director of the UCL Centre for Nerve Engineering. So UCL has a lot of neuroscience going on, and it also has quite a lot of engineering. So the Centre for Nerve Engineering was our attempt a few years ago to try and pull together the relevant neuroscientists, engineers and clinicians to really try and bring a multidisciplinary approach to bear on some of the problem of nervous system injury, along with our partner hospitals. And we kind of have this pipeline where we've got people, and there are people in my group working at all these different levels, from mathematical modelling through lab models into clinical research and reaching through into impact for patients. And I could talk about Centre for Nerve Engineering a lot, but I won't. I want to kind of move on. The other thing, as Chris mentioned, and this is my declaration of interest, is that I've been through the process of trying to spin out a company in this space, which is still at a very early stage, but I'd be happy to chat about that to other academics thinking about the same. So what I'm going to try and talk through today in a relatively short time is to try and give a flavour of some of the different things that I've been involved in this field recently. So the first one is really what I'd call the classic tissue engineering approach, trying to build artificial nervous system tissue that could be implanted to fill in gaps where the nervous system needs to be repaired. And actually, the most translational part of that for us is the peripheral nervous system, although this concept could be used anywhere in the nervous system. I'll then talk more about some specific projects around cell transplantation for Parkinson's disease, trying to use materials to treat traumatic brain injury, and then some more general concepts around delivery of factors and materials and molecules to promote regeneration generally. So just for anyone that's forgotten what peripheral nerves look like, they're relatively simple structures, just long, thin axons radiating out from the CNS, controlling all of the muscles, bringing back all of the sensation, always surrounded by Schwann cells, sometimes myelinated. And injury to these nerves, because they run all over the body, is really common and really debilitating. So it tends to be traumatic injury, tends to be younger people, often road traffic collisions, healthy young people going about their lives, all of a sudden have an instantaneous life-changing injury where they may lose complete function in a limb because of a peripheral nerve injury. Now, nerves do have some capacity to regenerate if you give them the right environment. So if a nerve's been cut, at the moment, the kind of clinical standard of care is to try and bridge the gap, repair the gap. If you can't pull the nerve back together and stitch it directly, then you can bridge it with a graft of a bit of nerve from somewhere else in the patient's body. And effectively, if you present the severed axons with an environment of like aligned Schwann cells, then they will tend to try and regenerate through that. It's still not great. And also, the thing that's really not great about this particular approach is that you have to destroy a bit of healthy nerve somewhere else in the patient's body where they weren't injured, just to get that bit of tissue you can use to maybe repair a more important one. So that's a big challenge, but it's also quite a nice opportunity for tissue engineering, because that's a relatively simple structure. That lump of donor tissue that gets taken from to be used in a nerve graft is simply aligned Schwann cells in an extracellular matrix that's aligned and has the right kind of biomechanical properties. So as a tissue engineer, my lab's been attempting to mimic that, And we developed this thing called NGNT, engineered neural tissue, which is effectively aligned therapeutic cells. So those therapeutic cells could be Schwann cells, but they're mimicking whatever it is the Schwann cells do to provide that nice environment in an aligned collagen matrix. So peripheral nerves are predominantly type 1 collagen fibrils, nicely aligned. And the idea is if we can implant this into a gap, then the severed axons will be able to grow through this implanted tissue, successfully get into the denervated downstream tissue, make their way back to the muscles and to the skin and to the targets. So we've tried various ways of making this, and we're not the only people to try and make an aligned cellular material to bridge gaps in nerves. But I think we've come about it in quite a biological way rather than more of an engineering way, which a lot of tissue engineering tends to involve. And this is the latest way in which we construct this. So effectively what we can do is make collagen gels, and when we're casting the gel, before it sets, we'll mix the therapeutic cells with that. And then what we're doing, and you can see in the video over on the right, is if you then suck that gel up into a cannula, you can see the drips coming out. Effectively what we're doing is making it much more dense. So this is a cellular collagen gel that's being squeezed, the water's being squeezed out of it. So what you end up with in the cannula is a very dense collagen gel, and here you'll see it pop out. So that is now a stable, dense, highly aligned tissue-like bit of engineered neural tissue. And if you look inside it, you can see aligned living cells, and it actually looks and feels very much like nerve tissue and is really rapid to make. We've had various other ways over the years of constructing this that take a lot longer. So I'm quite pleased with this approach because it's rapid manufacturing, kind of biofabrication. Now, where do you get the cells? That's been a question for us over a long time. When we first started out, we were using Schwann cells, but actually it's very difficult to find spare Schwann cells in a patient without damaging a donor nerve, which we want to avoid. So over the last decade or so, we've gone down a similar route to a lot of people who work in cell therapy and regenerative medicine. We thought, okay, we'll get stem cells out of patients, out of their adipose tissue, maybe out of their dental pulp, all these different places you can get stem cells, differentiate them to be Schwann cells, and then build our tissue and then implant it. Now, the problem with that is that takes quite a while, okay? To extract stem cells, culture them, expand them, differentiate them, build the engineered tissue is going to take a few weeks. Now, people don't know a few weeks in advance that they're going to have a nerve injury. So really, that's not a viable proposition for this. It could be in some circumstances, but for trauma, you really need to repair that nerve straight away with something you can get hold of quickly. So really, more recently, I've been much more interested in using allogeneic cells, so cells that come from another person, so that this could be prepared and ready and frozen and stored in hospitals and could just be thawed and implanted immediately on demand. And we've tried various different sources, and this is kind of the latest work we've been doing on this, is trying to use allogeneic cells for this. There was a proof of concept paper that we published a few years ago now where we used a source of cells which were human neural stem cells that had been used in other cell therapy processes. They were cells from a company called Reneuron who used them in a stroke trial. And we were able to use those cells in our engineered tissue. And we tested it out in a rat model of peripheral nerve injury, critical size gap over long time points. And there's some very boring looking graphs here, but that's good because they're all the same, because effectively what we're doing is showing equivalence to the gold standard autograft in our histological and our functional outcome measures, which is really reassuring because if it's equivalent to the autograft, then it means it could be used instead of the autograft without all the limitations of having to harvest tissue from elsewhere. So having had these results, that's why we then decided the next stage of this would be really a commercial opportunity rather than an academic-led thing. So we founded this spin-out company called Glialine. The NGNT will be an advanced therapy medicinal product. That's how these are regulated as an ATMP. And we're currently fundraising to support the kind of final preclinical work and GMP manufacture and this kind of thing. We've been trying to fundraise for a couple of years now, and actually I'm kind of in the process of perhaps pulling this back into the academic lab to move it a bit further forward, because there's a bit of a reluctance for people to invest in this area. But I think there's a reluctance to invest in anything like this at the moment. So that's where we are with that. I don't actually think that these kind of cells that we're using in this are likely to be the best thing possible. So in my academic lab, we're also exploring what other sources of cells could be used in the longer term future. And I don't think we're there yet, but the kinds of things we're looking at are, can we use induced pluripotent stem cells and turn them into Schwann cells? So this is some NGNT made using iPS-derived Schwann cells. And if anyone's ever tried doing this, you take them through a Schwann cell precursor stage before you get them to be Schwann cells. And actually, we tried implanting the precursors and the Schwann cells, and you actually get quite good results with the precursors as well as the Schwann cells. So it could be that that's something worth exploring as well. So this is one approach. But I think one of the other interesting things that's happened in the field recently, when I say recently, over the last 10 years, is a lot of groups have been looking at the role of endothelial cells after nerve injury. And one of the things that happens after nerve injury, one of the first things that happens in some of the scenarios where you get good natural repair with fairly small gaps, is actually endothelial cells migrate in, form structures, Schwann cells then migrate along those structures, and the regenerating neurons then migrate along the same structures. So actually, there might be a nice tissue engineering opportunity in engineering the endothelial cell structures rather than the Schwann cells. So that's what we're exploring at the moment. These are engineered neural tissue constructs made using iPS-derived endothelial cells, and I'm hoping to be able to start to test those out to really understand. I suspect, as is always the case with these things, it'll end up needing a complicated combination of different cell types to really get optimal regeneration. So in amongst all the experimental stuff, I mentioned earlier we also do some mathematical modeling. And I think that's an important thing just to mention. So I always include this slide these days because I've actually found it hugely beneficial to work really closely with mathematical modeling colleagues. So they call me an experimentalist, which I think is derogatory in mathematical modeling circles. But effectively, even if you ask a simple question like how many cells should we stick in, I used to put in a million cells per mil because that was just a good round number. But I don't know whether that's optimal. If I put too many cells into my construct, they'll use up the oxygen and they'll die. If I put just the right amount in, they'll use up the oxygen and become hypoxic and give off lots of really useful factors that will help infiltration. If I get the density too low, then you might as well not have the cells there at all. But how on earth can you work out what the optimal density is experimentally? That would be a horrendous number of experiments. So what you can do is quite simple in vitro experiments, give the data to the modelers, and they then generate lots of multicolored images, and they come back and say things like, James, don't use a million cells per mil like in your starting gel. What you need actually is this amount. And don't just put them everywhere. Maybe make a gradient, have more at the edges. And so actually you can, in silico, really kind of do some really quite exciting work around that. So I'm going to kind of move away from the nerve tissue engineering now. And in the next bit, I'm just going to mention a few other discrete projects. So this is a project which is still about cell transplantation, but this is now in the central nervous system. And there's a lot of work, including clinical trials at the moment, for Parkinson's disease, where people are doing cell transplantation. And one of the challenges with that, and there's a lot of challenges with it, but one of them is at the moment, the patients have to be immunosuppressed for at least a year. And I think there's kind of different opinions about that. But one of the things we wondered is, well, actually, there's only one small area of transplant here. And the problem is having to immunosuppress the whole patient. So are there ways of just kind of protecting those cells from the immune system locally? Now, people have done things like encapsulation of cells for cell transplantation in places like the pancreas. So what we've been developing is ways of trapping the cells in an alginate hydrogel in little beads. And then what that does is kind of provides a physical barrier that stops immune cells infiltrating. And these cells can still release things like dopamine, and they can release growth factors and that kind of thing. And we can also use the actual immunosuppressant, the tacrolimus, that's often used in the clinical trials, but in a controlled release manner. So you can trap the tacrolimus in microparticles and include that, and then kind of have this biomaterial encapsulation that's giving off the local immunosuppression. And in vitro, it seems to show some promising things initially, where we can reduce the T cell response to mismatched cells, and we can improve the viability of the cells, and T cells aren't getting to them. There's been some early work looking at this in the brain, in mice, and I think it's going to be quite a long journey before we can really kind of make this work. But I wanted to mention it as the kind of use of this kind of engineering technology. Another project at the moment, which is something which has been submitted but not published yet, is the idea of using these ECM biomaterials. So if you take a bit of tissue and kind of digest it, remove all the cells, you just get the extracellular matrix. You can effectively turn that into a biomaterial, and it's quite rich in different kind of signaling molecules and proteins, and maybe even vesicles and things like this. And we did some work trying to use, we started off trying to use the hydrogels to deliver some stem cells in a traumatic brain injury model. But we had some different groups in there. We kind of had the hydrogels on their own, and the stem cells on their own, and the combination. And it was really interesting. This is just a snapshot. We've got a lot of data around this. But really what it looked like was that the combination wasn't the best thing. The stem cells seemed to have a really positive effect, particularly on the astrocyte response. But the material itself, which we thought was just a vehicle, seemed to have quite an effect on what we think is probably the microglial response towards the beginning. And both of those things improved functional recovery on their own, not even with the combination. So it's reasonably complicated and raises more questions than it answers. But I think this kind of thing is really interesting because you know, we work with the neurosurgeons who are treating traumatic brain injury every day, and they're really limited in terms of the things that they can actually do for patients. So things that can potentially be added at that point of intervention that would improve the outcome is really valuable. So in the last few minutes, I want to talk a bit more generally about nervous system regenerative medicine. I think Divya gave a really good kind of introduction to the session. The idea that really we've got some opportunities and we've got some tools here. And while the more we get to learn about fundamental neurobiology, the more ready we're going to be with these tools to start to generate interesting new interventions. So fundamentally, if you're looking at regenerative medicine, when you can generate a pro-regenerative environment, it's generally through cells releasing signaling molecules, growth factors, cytokines, whatever, complicated mixtures of these, which have an effect on another cell and cause that cell to change its gene expression, to change its behavior, to regenerate rather than not to regenerate. So these are the kinds of opportunities and the kinds of targets. But this is really complicated. There's so many different factors. There are so many different downstream signaling pathways. And really what we have to, you know, there's no choice in studying this but to be quite reductionist and to try and pick one at a time and this kind of thing. And traditional medicines tend to pick one target at a time as well. And I feel like with some of the advanced therapies and some of the tissue engineering approaches, we're probably hitting multiple targets. You know, we're putting a cell in that's releasing a whole cocktail of factors, or a material that's hitting a load of things, but it's not very well understood. So there's kind of an opportunity here, but also a big challenge around how do we start to unravel what's really going on? When I say this material was effective, these cells helped regeneration, can we really understand what the signaling is that's going on? Because then perhaps we don't have to do this crude thing of putting in living cells. Perhaps we can put in molecules which will trigger the same responses. So I think there's a load of opportunities there. the community's got a toolkit of technologies, and I'm not going to talk through these, but I really talked a bit about, hydrogels, fibers, particles. All these things can be used to make structures and also used to do really controlled local delivery of different factors and different molecules in a spatially and temporally controlled way. So I think there's a lot of tools here and a lot of opportunities. And what would be nice would be to try and join the fields up a bit more in a way so that we can kind of really use the neuroscience knowledge to improve these. So without knowing a huge amount about it, we can still do some things. So this is some work that was published just a couple of weeks ago from our lab, where effectively we were making nanofibers that would release a growth factor, GDNF, which we know is really helpful in this kind of regeneration. And this is in vitro at the moment, but effectively having aligned fibers versus random fibers really improved the growth, and having aligned fibers that release GDNF improved it even further. So the postdoc who's working on that has now tried kind of making these flat sheets of material into this sort of Swiss roll arrangements with multiple layers, and these have been through the rat model now and are being analysed at the moment to see, to really understand what's happening here. Because, of course, if you start locally releasing growth factors, that can sometimes be a bad thing because it might encourage regeneration towards the growth factor and then halt it at that point. So you really need to control it so you get good growth through. And then the last thing I'm going to talk through is targeting a particular downstream signaling pathway. So this is some work I was involved in. Effectively, some other people at UCL who work on cancer were really interested in this PI3 kinase pathway. So PI3 kinase signaling is one of the things that sometimes goes wrong in cancer. So a lot of people have worked on trying to inhibit the pathway. These guys came to me and said, James, we made an activator of this pathway. Would that be useful? So we thought, okay, we'll test this out. We put their small molecule activator on some neurons in a dish, and really saw a very nice dose-dependent increase in neurite growth. And we then took that forward into the rat model. So look away if you don't want to see rat microsurgery images on the screen. Effectively what we did is a sciatic nerve crush, and we then administered the drug in various different ways. Not a drug, it's a compound, either by injecting it or with osmotic mini pumps. And then we looked at various downstream places, and what we saw was an increase with our compound in things like the electrophysiological response, the number of motor neurons, and that correlated very nicely, and it's always quite nice when your histology correlates with your function. And also we looked at neuromuscular junction re-innervation, and that correlated with the improvement in motor response. And interestingly, when you look close to the crush site, so this is kind of functional recovery down here. When you look close to the crush site, really you see no effect, which is reassuring because it suggests that what you've done is just accelerate the natural regeneration rather than create a load of sprouting or something like that. So where I want to finish is just by mentioning that I've also, in the last few years, really enjoyed some of the engagement activities we've done, really kind of reaching out to clinicians and people living with nerve injury, because as we're trying to translate these things, we need to make sure that we've got good clinical outcome measures that can be used to evaluate some of the new technologies, and also finding out what's meaningful for patients. So we were recently involved in one of these priority setting partnerships, which really helped with that. So I'll stop there. Acknowledge the people that did the work, and the collaborators and funders. I've got a website. I'm not very good on X, but feel free to follow me. And yeah, just really open to collaborative opportunities and that kind of thing. And I'll just leave you with the current projects in the lab at the moment. Thank you.
Have we got a couple of minutes for any questions, if there are any? Have we got one?
Yeah, thanks, that was amazing. It's more of a logistical question, really. So when you extrude the gel from the cannula, obviously for GMP, if you've got cells in there, sterility is going to be a big thing. How do you overcome that? Because you obviously can't just
Yeah, yeah. So, so I think in terms of transfer to GMP, effectively what you would do is the whole thing would be done in a clean room. So you'd have a GMP cell bank that would effectively be in the clean room. All of your materials would be kind of GMP validated. And you can put that extrusion apparatus, which is a grand word for it. It was actually a 3D printer that we took apart. But you can put that inside a hood. And actually, I like the idea that, so I think for our first in human, we're looking at kind of doing this in a semi-manual way in a GMP clean room with a person actually kind of pushing the button and then kind of moving it and wrapping it up and packaging it. But it kind of lends itself, because it's an extrusion process, it kind of lends itself to full automation, I think. So I think it could be scalable and could be something where actually it just all happens inside a sealed kind of machine without much other intervention. But that's a longer term vision. But actually, I mean, you asked specifically about sterility. So part of this, I think the way it would work is we'd make a batch, and then some of that batch, in each batch, would go off for kind of release testing and sterility testing and all the same sorts of things that a cell therapy would have to go through.
We've got time for one more, if there is one more.
Very nice question. I didn't see that much clinical physiology to functionally measure your treatment. Because sometimes you inject 1 million, 5,000 K neurons, they are all there, but the problem is that only 20 of them make synaptic connections.
Yeah. Yeah. So you mean for the peripheral nerve? Yeah. So what?
Or axon.
Yeah. I mean, I agree, and actually, it's really important to think about because what we tend to do is look. So our electrophysiology tends to look at compound muscle action potential, which I mean, it's not really measuring individual axons in any way. You're measuring kind of motor units. And actually, that top left graph, the motor unit number estimation, is quite a good way of you can kind of correlate that. When you get an increase in motor number, that's like motor unit, you can correlate that with the kind of number of actual myofibers that have been re-innervated. So it's quite a reasonable way of correlating it. But in terms of functional recovery, whether that's meaningful to patients, whether it's getting back towards control. I mean, here we're just comparing it to a vehicle. It's considerably lower than a healthy nerve would be. So in some ways, it would be really important to kind of, all right, we might be able to double our re-innovation, but if that's still kind of 10 times smaller than healthy nerve, then maybe it's useless. So I think these are important things to think about.
Thank you. I think in the interest of time, we'll move on, but James will be, well, we'll all be around later, so if you've got any more questions.
I'll be around. Thank you.
So yeah, I think next up is Stuart Jenkins. So I've known Stu for a long time through our work at Keele. He's a lecturer in neurobiology, School of Medicine at Keele University, and he's done lots of really nice work on microglia and their interactions with neural tissue engineering strategies. Stu?
Thank you, Chris. Yes, good morning all. My name's Stuart Jenkins. I'm going to talk about biomaterials and microglia and why simulating the brain immune response might be particularly important for neural tissue engineering. The clinical need should be relevant to everyone in this room. Neurological disorders are costing upwards of $1 trillion a year, and this is comparable to the costs on heart disease, cancer, and diabetes combined. And global disability by 2030 is being estimated to be 50% due to brain disorders. All this makes neurological health a global public priority. And we're going to need targeted interventions to reduce both the personal and societal burden of these conditions. Restoring neural function, there are being particular progress in recent times using neuroprosthetics and brain-computer interfaces and things which are basically bypassing tissue problems, but there's still a patient preference to regain the intrinsic function within their neural tissue. And so we want to see whether we can go beyond these assistive technologies and get to the restoration of actual functional central nervous system tissue. With this in mind, it's been already mentioned by Professor Phillips, but I'm also going to talk about some of the Versatile platforms we've got for neural engineering, nanoparticles, nanofibers, and hydrogels. And I'm going to talk about the neural immune responses in particular to these and why they might be critical to the efficacy and safety of all of these platforms. The microglia are the brain's immune component. They're the principal immune cells in the central nervous system. They are highly active cells, are constantly surveilling their immediate environment. And the video I'm going to play on the right in a moment, you will hopefully be able to see the territorial nature of many of these cells. They'll tend to sit in one particular spot, extending and retracting membrane as they explore the immediate environment. You'll also see a cell undergoing phagocytosis. The blue marker there will be the second cell of interest to have a look at. But the first one I'm just going to play is the bottom right patrolling one here. So one of these microglia is off on maneuvers. It's exploring its environment, in contrast to most of the microglial cells, just sitting in one particular spot. And there's now phagocytosis happening there. One cell has lunged down and engulfed another. So these are exhibiting the rates of activity of microglia compared to the other immune cells. In this polyglial culture, there's a bed of astrocytes at the bottom. Microglia and oligodendrocyte precursors are sitting on top of those cells. So that's the glial cells compared with each other. Surprisingly few biomaterial studies are really looking at what microglial responses are. This is mainly because many neural models lack a microglial element. Plenty of test beds of biomaterials don't comment on what microglial responses are or might be. Co-culture systems derived from induced pluripotent stem cells or neural stem cells, for example, don't immediately endogenously create microglia. They're from a different origin. Early in development, microglia arise in primitive hematopoiesis, invade the developing central nervous system tissue. The blood-brain barrier forms around them, and then microglia grow up as kind of cousins to the rest of the neural cells. And also what cell lines we have for microglia frequently lack some of the critical immune responses, or at least have modified immune responses. We've developed a variety of neuroimmune relevant models, both normal and pathological tissue being simulated. And I'm going to start by talking about nanoparticles in these systems. So nanoparticles, they're highly versatile. A whole variety of applications are available, including drug delivery, genetic engineering, and labeling of cells with particles such that they can be tracked post-transplantation. But how do microglia respond to nanoparticles? Here's a test nanoparticle, one of the many we've used. These are called spheroparticles. They're about 360 nanometers in diameter. They've got a fluorophore stained core and then an iron oxide ring around them. And if we incubate these particles with astrocytes, here's a pure astrocyte culture, we'll see that astrocytes will take up these particles. You can see there's heterogeneity in how much particle uptake is per individual cell, but these cells clearly take up these particles. If we do the same thing with a pure culture of microglia, again, we can see these particles are taken up even more avidly in microglia than astrocytes. But we've looked at whether you can anticipate this astrocyte uptake would be replicated in an in vivo situation. And so if we do a defined co-culture here, we've combined 50% astrocytes with 50% microglia at the bottom left. And now the green GFAP positive astrocytes here are showing barely any particle uptake due to the presence of microglia. The microglia are not stained here. The red channel is dedicated to the particles rather than staining the cells, but you can see that microglia, the GFAP negative cells, have engulfed the majority of the particles, depriving astrocytes and suggesting that the prediction you might make from the first culture there, that astrocytes would take up these particles, is unlikely to be carried out in vivo, and so there may be limited predictive value in assessing particle responses in a monoculture. We've just quantified levels of uptake here, so the far left bar in this graph is just showing astrocyte uptake in a pure culture, and then the second bar from the left is the astrocytes with microglial presence, and there's a dramatic reduction in particle uptake. Observationally, if we've even got like 5% microglial contamination within an otherwise pure culture of other cell types, we'll see that the microglia dramatically diminish uptake by the non-microglial cells. This represents a significant extracellular barrier to particle delivery. We've also observed these competitive uptake dynamics in real time. So I've got a video here, the same polyglial culture, a bed of astrocytes with oligodendroglia and microglia on top. The microglia have got M label in them, and as these particles are introduced, you can see the microglial cells hoovering up these particles, engulfing them rapidly. Very few particles end up in non-microglial cells, and you can actually see the sweeps of membrane from the microglia begin to diminish as the microglia engorge themselves and become completely full of particles. They lose spare membrane to start reaching out, and they become relatively static and bloated. So we've also gone and looked at whether this would occur in an actual pathology situation, an actual model of injury. So here's an organotypic slice culture system we've developed for spinal cord. So we can take sections of spinal cord about 350 microns thick, a third of a millimetre. We can then culture these at an air-medium interface and induce mechanical injuries into them. Here, a transecting lesion. The bottom left, the green image here, is showing high viability. So live cells are in green, dead cells are in red. We get high viability in these slices, high preservation of cells. And then when we induce this lesion, we see that cells will also move into that lesion and start some regeneration. So this is a few days post-lesion. And on the right here, you can see blue DAPI staining showing that cells are all present. And the red here is nanoparticles introduced into that lesion site within an organotypic slice. Here's another video. So this is a close-up of one of those lesions. The white dashed line there shows the edge of the lesion with the intact tissue above, the lesion below, and we observe that microglia move into this lesion site, and we introduce the particles, and we see the same effect of microglia hoovering up the particles. It's incidental to this, but the white arrows just show 3 mitotic microglial cells within this. But microglia are again dominating uptake versus other cell types. We're pretty happy with our identification of microglia based on their very characteristic rates of movement and behaviours, but we have also managed to track these cultures post-video through fixing to immunostaining and demonstrate that these are definitely microglial cells. So if we follow these up, we can track the same cells taking up the particles as being categorically microglial. Okay, so microglia are dominating uptake. That may not necessarily be an issue. If you just need your particles to be inside any neural cell, maybe that's not going to worry you. But we wanted to look at what these consequences of microglial domination of uptake might be. Here's our same spheroparticles again. There's a polystyrene core with a dye, an iron oxide surrounding the particle. Transmission electron microscopy of these particles shows a faintly grey central core with a ring of iron oxide around it. So the dark particles here are iron oxide particles and a ring around these sectioned particles. And if we look at astrocytes under TEM, transmission electron micrographs show that these particles accumulate within the astrocytes and often perinuclear in these accumulations, but the particles seem to be the same as they were when looked at without cells. They seem to be intact. If we perform TEM of microglial cells having taken up particles, we begin to see that the particles no longer have this, Chris might be a bit much, but this clear ring of iron around the central core. The particles seem to be degrading, leaching ions. We seem to be degrading these particles within inside microglia. And here is a close-up of a lysosome within a microglial cell. And hopefully you can see that these particles no longer have a clear ring of iron. These seem to be degrading particles, and the leaching of iron from these particles might well lead to toxic effects. Loose iron within cells increases what are called Fenton reactions, and we wonder if this may be a mechanistic explanation for why there is a drop-off in microglial viability, with higher concentrations of these iron oxide particles. So we need to assess whether biomaterial breakdown by microglia might lead to toxic byproducts, and we need to look at whether this combination of microglial uptake and breakdown may be a major challenge to targeting nanoparticles to other neural cell types. Taking that as a likely hypothesis, what can we do to mitigate this clearance of particles by microglia? One of the strategies we've looked at is putting stealth coatings on the nanoparticles. So at the top left here, we've got a cartoon of a nanoparticle. This one's got an iron oxide core rather than an outer ring. It's got a fluorescent layer, and then there's a dextran coating. Dextrans are commonly used pharmaceutical ingredient for drugs, tablets, etc. So we've got a dextran coating there as our control particle. But if we use an identical particle with instead a polyethylene glycol coating, PEG, PEG is a hydrophilic coating, and it's used to improve drug bioavailability. It hopefully helps to stop protein aggregation around particles and may reduce immune clearance. We've also characterized these particles by FTIR just to confirm the surface coating. At the bottom left, we've got our controlled dextran particles incubated here with microglial cells, and you can see dramatic uptake similar to the spheroparticles we showed previously. Now, once we compare this with the PEG coatings, we see that microglia will still take up these stealth particles, but there's a notable reduction in the amount of particle uptake per individual cells, although, as you can see in both pictures, heterogeneity in levels of uptake for both types. Quantifying these, if we normalise it to the dextran, we can see there's a measurable reduction in particle uptake with the stealth coatings, but they do not completely evade immune uptake. With our other particle studies, we've recently been working with a protein co-crystal. This is it's an insect virus, actually produces this protein which self-assembles into these dramatic and impressive cuboid morphologies. And so if you can co-crystallize these with a drug protein, or in this case for testing purposes, green fluorescent protein, you co-crystallize polyhedron with a drug delivery protein and you get slow release of this drug. This may have applications in the central nervous system, so we wondered what microglia would do with these particles. So hopefully you can see the really quite impressive cuboid squared corners on these particles, and we've measured the edges of these. They've got nano scale for some of them, but many of them are getting into several microns in size. Incubating these with our microglia, we do see uptake of these by microglial cells, but we also see, even after 24 hours, a time period at which we would see almost all the other particle types we've looked at cleared and engulfed by microglia. We see residual PODS particles remaining unendocytosed, and we're wondering if there may be something around the geometry of these particles where the microglia are not as interested in clearing them. Okay, moving on to nanofibers as a second topic. They've already been mentioned, which is helpful for me, but nanofibers can be used to span lesions, bridge gaps. They may be used to induce migration of precursor or stem cells into lesions and promote regeneration. They've also got a particular application for guiding axonal nerve fiber growth across lesions. We're still hoping it's synapses at the other side, but it's a strategy. They also seem to have some axon mimetic properties at times. There's people reporting that they can culture oligodendroglia with these, and they find that they are simulating myelination of nanofibers, so there may be scope for studies there. We've looked at cell-fiber interactions, and I'm going to talk about the microglia element in particular. So here are two of the scaffolds we've been experimenting with. The top left in the red box, we've got a mat of randomly oriented fibers together, and on the right in the green box are some highly aligned parallel fibers. We've analysed the directionality of these, and we see that the aligned fibers have got a much narrower window of direction compared to the broader range of direction in the random. And these particular fibers are about 900 nanometers in diameter. And when we add microglia to them, the microglia will adopt the directionality of the aligned fibers, measurably greater than on random or a glass cover slip with no directional cues. And if we look at these in higher magnification and go and look at the cells in particular, the cells tend to become bipolar. They'll tend to elongate along these fibers. And this is a measurable increase in the aspect ratio. If we take the length of the cell, divide it by the width of the cell, so that a perfectly circular cell would have a value of 1, we see an increased aspect ratio, an elongation of the cells on these aligned fibers. This alignment we've also looked at in a simulated injury model. So if we take similar fibers, but this time these are polylactic acid, PLA, rather than PCL, which the previous ones were, we can put these into a windowed frame here so that the fibers are spanning the gap. These particular ones have also had a red dye incorporated, rhodamine B, so we can see them fluorescently labelled. We can then overlay this over on the third image there. This is another one of our spinal cord injury slices. So we've got a lesion across the middle, and we've placed fibres across that lesion. And microglia exhibit the same association with fibres and bipolarity and elongation within it. So we're seeing the same microglial behaviour in a simulated injury with central nervous system tissue. So they're lining up with the fibres, but what are they actually doing there? We've developed capacity recently to do live imaging of these cells on fibres. So here we have some polycaprolactone fibres. These have been generated in the lab, and they're across an O-ring, so just a circular version of the previous frame. And we've applied microglial cells to these fibres. And if we observe these fibres, we see that they become bipolar and elongated again. And when I start this video in just a moment, the first cell I'll draw your attention to is this one. This cell exhibits the greatest rate of movement on these fibres. So if I just play that there, this cell here, it extends off to the right, and then it's actually going to ping back leftwards at quite an alarming rate, like an elastic band or something. So at the moment, we're characterising what kind of rates of movement we get of these cells on the fibres and what they do for overall interactions, but we see the same kind of territorial responses. We've got this elongation, the cells are clearly extruding and retracting membrane, but when they meet neighbours, they'll tend to retreat from them. It's fewer than 10% of the cells do we see one cell pass another cell and go beyond it. If they meet another cell on the fibre, they halt in their tracks and back up. They don't go past each other. They hold this territoriality. They're exploring, constantly extending membrane across the fibre. We've recently, just too soon to properly squeeze into this, got some scanning electron microscopy of these cells on fibres, and we can see they really quite tightly associate their membrane with the fibre in places, and we're looking at understanding at a very high resolution what they are doing when they meet the fibre. Given the uptake and degradation of nanoparticles, we don't yet see any obvious degradation of these fibres. We don't see them chopping the fibres into bits, we don't see breaks in the fibres, and we don't see, so far, fibre fragments inside the cells. So we also wonder if there's something geometrical about nanofibres which maybe prevents or never even triggers a phagocytic response in microglial cells. Okay, I'll finish with a little bit on hydrogels as well. Variety of clinical applications. I don't need to labour the point to this audience. You've already heard excellent stuff, but drug delivery, protection of cells post-transplantation. There's also in vitro applications. We're interested in using hydrogels to create a neuromimetic substrate so that cells will behave more realistically in the laboratory, both for mimicking normal and pathological tissue. And we can induce pathological features within hydrogels. So what do microglia think of hydrogels? Here's microglial morphology. First of all, on the left, on a standard glass substrate, it's on a glass cover slip, 2D surface here, 2D hard surface, you get these flattened amoeboid shapes, almost kind of fried egg morphologies. On the right, we've got microglia in a hydrogel. So it's 3D, this is a collagen hydrogel. The cells are highly branched here, and I would suggest that the morphologies over here more closely resemble this histological section here, showing highly ramified microglia from a tissue section. So that's morphology in the first place, but how about microglial behaviours within a hydrogel? Here are more microglia on a 2D glass slide, and on the right we've got microglia in another collagen hydrogel. And just as a reminder of in vivo morphology at the top right there, I'm going to play these two videos. Microglia here are flattened, rounded, large sweeper-large skirt of membrane around a lump where the nucleus is. And if you compare that with the movement on the right-hand side, you can see that there's some kind of territoriality in both cases. Many cells will stay in one place, but also others will go wandering. But hopefully you can see the highly ramified morphologies prevalent within the hydrogel compared to the more flattened morphologies, perhaps one process, often a kind of tail left behind in the 2D environment here. It's almost a kind of a stingray morphology at times, where there's a tail left behind and a big skirt of membrane as the leading part of the cell. When I'd first seen these, I imagined that single process was exploratory and leading. It seems to be a tail. Okay, we've also developed injury models for hydrogels, so I'll finish with just one of our more advanced models in the lab. This is just showing the multiplicity of cell types we've got present here. So we have astrocytes, oligodendroglia, microglia, neurons, and oligodendrocyte precursors, all within a single gel. You can only stain so many cells at once, unfortunately, but here's a 4-channel stain showing multiple cell types altogether, and the morphologies here are all very different to in a 2D substrate, and hopefully more in vivo-like. And we can use a facile method of inserting and twisting a pipette tip to induce a puncture-type injury within these gels. We find this is quite reproducible, it's quite simple to do. Once we've induced this injury in these multicellular models, we notice that microglia, astrocytes at the edge of the lesion site will tend to upregulate GFAP and present various features reminiscent of glial scarring. This is in particular comparison to more distal astrocytes elsewhere in the gel away from the lesion. And if we look at microglial behaviours, again, near the lesion edge, where we've caused this simulated injury, we see more amoeboid microglial morphologies compared to distal parts of the gel where the cells are more ramified, and we've quantified this. Now, having induced the injury and observed pathological features, we can then also take biomaterials. Here's an example of a surgical-grade dural sealant called Duragens. This is used to patch up penetrating injuries to the meninges. We can take this bovine collagen surgical-grade sealant and we can implant pieces of it into these lesions, and if we study the microglial responses, other cell responses have been studied, we can see that microglia will invade into these implanted collagen plugs of hydrogel. And we found this quite reproducible, which is all the graph on the far right is showing. So that's my whirlwind microglia responses to biomaterials and some of the models which we are using. So I will thank the rest of NTech and everyone here, and I'll take any questions you might have.
We got time for a couple of questions?
So it was a great talk, thank you. So I was wondering about the, thank you. So I was wondering about the amount of nanoparticles that the microglia can get. Is that like, is there a limit, or the cells explode, or I don't know if you mentioned that or not.
We haven't seen explosion yet. We see a kind of almost like a stasis. We probably need to leave them longer and see what actually happens. But they do seem to reach a kind of maximum level of uptake when they fail to have any more membrane sweeping out. It's a slightly difficult thing to fully quantify. But from the video evidence we have, by the time they fill up and you can no longer see bits of membrane leaving the cell and sweeping out, once they're kind of engorged there, we don't tend to see increases in particle uptake there. Difficult to categorically say it, but I think they run out of capacity by the time the membrane can no longer extend outwards.
Thanks very much. That was really interesting and lovely videos. Just really briefly to ask about the microglia source. Which type of microglia was it?
Beg your pardon, I did never mention that.
Well,
Yeah, a couple of those were rat.
Some limitations of those, the sort of ones you buy, but where were these ones from, sorry?
Yeah, so the majority of these are mouse. There's probably one or two older ones there, might well be rat. I think the spinal cord one with the nanoparticles might have been a rat, and all the rest would have been mouse. So these are early postnatal mouse would be the source of these microglia. I have also sourced adult mouse and adult rat microglia and managed to get those into gels and things as well, so we can get those. We haven't.
Are they always used freshly sourced? Are they?
Oh yeah, yeah. It's fresh from primary tissue for everything shown here. The majority of our stuff. We're looking at getting humanized models and looking at what human possibilities there might be for microglia, and we've briefly looked at them in a chunk of human cerebellum tissue, but we're not routinely getting human microglia yet, unfortunately, not primary anyway.
Okay, thank you very much.
In the interest of time, we'll
Yeah.
move on, but yeah.
Thank you.
Very much, Stu. So yeah, next we've got Marimelia. She's a professor in the Department of Biochemistry and also Vice Director for the Federal University of São Paulo. So yeah, Mari's done a lot of work on fabrication of tissue using 3D printing. We met last year or 2 years ago? 2 years ago, so we wrote a chapter in the same book, which is quite a nice coincidence. But yeah, thanks, Mary.
Yeah. Okay. So first, thank Chris and Divya for the invitation, and thank you for being here. It's been a great pleasure to be at the BNA. So what I'm going to talk about is the models that we have been developing to understand different neurological pathologies. So it's going to be, I was very happy that Stuart finished the talk talking about models that we can use. So I think we can have some talk after because the microglia is missing in our models. So I'm going to present work from these students of mine. And I have been interested in neurogenic niches for a long time. First, studying them in mice, especially looking at how the neurogenic niches responded to injuries, especially traumatic brain injury and stroke, as a model for acute injuries to the brain, and also how in neurodegenerative disorders such as Alzheimer's disease. So in all these diseases, the niches, the neurogenic niches, even in the adult, they will respond and they will produce new cells, neuroblasts, that will, in the case of acute injuries such as the TBI and stroke, these neuroblasts will migrate towards the injury site in an attempt to restore, the dead cells. But of course, in mammals, it doesn't work because otherwise we wouldn't have like sequela after like a traumatic brain injury. So as I said before, I was always interested in understanding, the mechanisms of generating new neurons in this type of place. And when I decided to kind of start moving from animal studies into more complex in vitro studies, I decided to focus on two main structures. One is the neurogenic niche, which I'm going to talk about today, and the other one is the blood-brain barrier that I don't have time to talk about, but if anyone is interested in BBB physiology or BBB modeling, I'm very, very much interested to talk about that. So when we look at the neurogenic niches, we can look at two different time points. So we have in the neurodevelopment, in the early neurodevelopment, we have a neurogenic niche that's going to be responsible for creating all the cells except for microglia that will form the brain parenchyma. And in the adult, there are some places in the brain that still keep these neurostem cells, the subventricular zone and the subgranular zone in the hippocampus. The mechanisms or the cell events that happen, they are very similar. If we look, from like an up look, we're going to see that there's proliferation, migration, differentiation, and some cell death. So this is what we are trying to look at. And why these two types of, or two extremes, I'd say, in time. So one is that in the early development, if neurons are not in the right place at the right time, we're going to have neurodevelopmental diseases or disorders such as autism spectrum disorder. And in the neurodegenerative disease, in the other scale in time, or when we age, these are also, so the source of new neurons can also be affected. So, and also these are two like big classes of diseases that affect humans. So as I mentioned in the beginning, I have been interested in studying neurogenesis, and first thing we did was studying in animals, and there are several types of animals that we can use. And this is just one example of a work published by our lab many years ago, in which we treated the animals. This is the double transgenic mice for Alzheimer's disease, and you can see that when you treat them with leptin, there's an increase in the number of cells. So it was a nice way to study neurogenesis. And then going to more complex settings or more complex in vitro models, there are several already models that have been used. So we have from the IPS, the organoids or the cortical organoids are maybe the golden standard these days for studying, especially studying neurogenesis. But we also have like traditional scaffold in which we can add cells that we already saw some of these type of scaffolds in the previous talks. Neurospheroids that are simpler, like the neurospheres that we can get from the neurogenic niche, the adult neurogenic niche. They are simpler, but they also have a great deal of information that we can get from them. And the 3D bioprinting, which uses hydrogels as the biomaterial, and then we combine this biomaterial with cells, and we can give them a shape that we want. Of course, when we go from the whole animal to these structures, we lose complexity, but we also can use human cells that we cannot study in the whole animals. I mean, in a whole person, of course. We have done some work in collaboration with a colleague from the Einstein Hospital in São Paulo, in which we combined organoids with 3D bioprinting. And so this was published already, and we were able to look at the organoids in immunofluorescence, and also to do some electrophysiology using MEA, the multi-electrode array systems. So you can see that the organoids, they are firing, and so the neurons are actually connecting and talking to them. So as I said in the beginning, the aim or the goal for this part of works in my lab was to mimic the one neurogenic niche using human IPS, the induced pluripotent stem cells, that we differentiate into neuroprogenitors. And then we are trying to see if, when we do that, when we bioprint the NPCs derived from IPS, if we can model the neurogenic niche. And what we are looking for is, if there is proliferation, if there is differentiation and migration. We haven't looked at death, but I believe that we can see death if we look for it. So the 3D bioprinting has three different types of bioprinting. What we use is the extrusion type, which is I think it's most common to use for living cells. And so what we do is that we mix a hydrogel with cells and then put this in an extrusion bioprinter. So this combination of hydrogel and cells, we call it a bio-ink. And the extrusion means that there's a piston that's going to go down, and then the hydrogel or the bio-ink, hydrogel plus the cells, will be placed on a plate. So this is just a photo of one of the printers we have in the lab, and this is a very simple example of how it works. And here are some constructs already. You see all these white dots here, they are cells that are already proliferated, and we have published a few papers looking at different compositions of hydrogels. This is one of the works we did. So there's a lot of characterizing the bio-ink, characterizing the hydrogel. And here in this paper, we did several different compositions for the hydrogel, changing the amount of gelatin and gelatin metacrillate, or GELMA. And we did all the rheology studies to see how stiff or soft the bio-ink was. And you can see that when we change the proportions of GELMA or gelatin, we really change the structure of the bio-ink, which is important for the cells. In that paper, what we saw was that we needed to add gel tracks, which is similar to matrigel. It's an extracellular matrix mixture. And when we did not add gel tracks, the cells did not survive a long time. And here you can see NPCs that were bioprinted and then kept in culture for 28 days. We can see GFAP and MAP2 showing that we have the presence of neurostem cells and astrocytes, because GFAP labels both of them. And also we did other cells such astrocytes, and they also go very well. We are not able to bioprint neurons that are mature already. They are very sensitive, and we could not do this. But astrocytes, they go well. And then, so after we published that, we start looking at in more details in what was happening in these constructs in terms of the cell composition and what the cells were doing. And that's when we really saw that this could be a model for the neurogenic niche. We start seeing structures like this that they are similar to ventricular zones. And if you look like here, and they start as a rosette, like this one, that is kind of closed, and then they open as if they were forming several ventricular zones here. If you look at cortical organoids, this is something that also happens. We can see the same type of structures in organoids. So another thing that we did in this constructs was to try to look at migration. And if you look at the blue dots, they are the nuclei that was labeled. We can see that the cells, they move. And another thing is that we saw tube-3 labeling showing that cells, they do mature and they become neurons. We don't know what type of neurons, and probably they are not specific. Maybe you have to change the media that we're using to induce proliferation, differentiation. But one thing that's very interesting is this is color-coded for the depth where these neurons and processes are. So blue and green means that they are more at the surface of the construct, but they also go deep into the construct. So to further characterize these bioink that we developed, so this was a development from the first paper I showed you. We did more material characterization of the cells. We studied degradation and how much swelling happened to the construct. And what we saw is that if the construct doesn't have any cells, it doesn't last like a week. It disintegrates. So it needs to have the cells, because probably the cells are making extracellular matrix that is putting them together. And this is, if someone is interested in more of the material characterization, we can talk about that later. Doing live and dead evaluation following the days, you can see that the cells, they are viable, they proliferate. And here again, what I showed in a previous slide, showing all these white clumps or like clusters of cells that are connected with each other. And when we did some testing with the medium composition, so this is what we call an expansion medium. So it has FGF and B27. And this one is a maturation medium. It has GDNF and BDNF and no FGF. So we kind of stop proliferation and then stimulate more differentiation of the cells. And we can see that in both, although they have different arrangements and different amounts of each cell type, but we do see in green JFAP in both and TUB3 in both, and also nesting that marks non-differentiated or neuroprogenitor cells. And we think that these are very interesting fibers. They might be radioglia type of cells. And in red here we have what would be like neuronal processes. And so we got a little bit better in recording the migration of the cells. So now I have one of my students who got these images. She's like looking at quantifying this, because we need to know, we want to know the speed of the cells and everything. So for the future directions in this part is that we are now looking at using ASD, or Autism Syndrome Disorder cells, and then to study how we can use this model to study, especially migration in this case, because it's one patient that has a reeling mutation. And for the next 5 minutes, I'm gonna just show you, we have been trying to model Alzheimer's disease neurogenic niche. So what happens in Alzheimer's is that the accumulation of A-beta or beta oligomers outside in the extracellular matrix may be triggering cell death, neuronal death. And so we wanted to explore that in this same type of settings. These were done with mice cells, but here we see, and these were from adult cells. So this is a neurogenic niche from adult cells. So we have neurospheres. They are not more complex because they come from adult animals. And what we saw was that an increase when we add A-beta to the bioink composition. We have an increase in the amount of stress or oxidative stress that happens, even after a few days. And this is the how the it's a scan microscopy of the construct with the neurospheres. And we are using these model. So this one is already published in the Journal of Bioprinting. And now we are using the same model with human cells, so NPCs derived from iPS. And we can see that they also suffer from when you add A-beta to the bioink. And it's concentration dependent, and here's like 8 days after bioprinting in the presence or absence of A-beta. And so we see that there's a decrease in the number of cells. We don't know if it's inducing cell death or just reducing proliferation. We haven't looked at that. We're looking at the viable cells at this time. But also we see an increase in the oxidative stress, which is another marker for Alzheimer's. So this is preliminary data, and it's still ongoing. And we have a collaboration with two researchers in University of Strasbourg, that they have neuroprotectant compounds that we're going to use to see if they can protect, the neuronal death. So this is the second one. And just for my final slide, I have one postdoc that is developing an application to help the selection of hydrogel composition. And this is just two screenshots from the application. So she took like 300 papers that had hydrogel compositions and viability of different cell types. And the application is intended so you can insert the the biomaterial you're using. You can insert the cell type. Here, like astrocytes, Schwann cells, different type of biomaterial, and also there's another function here that you can help us increase this database by inputting your own cell type and biomaterial. And she's right now doing the real validation of this application. But she did an in silico validation, so she took other papers that were not included in her database, and we see a pretty good correlation. So we hope that this will be a application that everybody that's interested in using biomaterials can use. And so that's it. I thank you very much for your attention. And I put this photo here because it's when Chris was in our lab. This is my group. And this is a bigger group we are part of. This is the National Institute for Science and Technology in 3D model that's hosted in my university. Thank you very much.
Thank you. Yes, we've got time for one question, I think.
Yeah, that's a really nice talk. You mentioned that you could control and you measure the stiffness of the materials, and you see it has an effect on the cells. And I just wondered, how does the stiffness compare to the stiffness of natural tissue in
the brain
development or in adults or in disease or different states?
Yeah. So we have one paper in which we did different stiffness and put astrocytes there, and we wanted to mimic the glial scar. So the glial scar is softer than the real tissue. And when you put astrocytes in this softer material, it reactivates the astrocyte, as it does when it goes to a glial scar. So here, what we do is for these we are not doing different stiffness for like early development or adult neurogenic niche, but we are doing the same one that is close to what you have in the brain, the undamaged brain. I really don't know the numbers for it, but yeah.
Thank you.
I think we're going to have to move on for time,
Okay, thank you.
but we're going to be around if there's more questions.
Yes,
Thank you, ma'am. Okay, and our final speaker is Ben Newland. He's a lecturer in Cardiff, and again has done some really nice work on biomaterials for neural regeneration. Thanks, Ben.
Thank you. Okay, hi everyone. So I think I have a little bit of time before you have a break, so I think mine's a little bit shorter, but yeah, thanks for the introduction. I'm from Cardiff University, and really our lab is focused around this question of how can we deliver drugs and proteins and even cells to one region of the brain, as I try to depict here. Okay, we're really interested in disorders such as Parkinson's disease and brain tumors, anywhere where there's a focal pathology. And actually, the development of the scaffold, which I'll talk to you about in a moment, have become used in other research labs as tools, basically. And so I'd like you guys to have a think if there's any kind of spatio-temporal effect that you want to achieve in tissue, and I'll talk to you about this word in the next slide. But is there some sort of inhibitor, some sort of protein or anything like that, or reagent or dye, that you're interested in getting to one area of tissue and not another? And that could be an organoid, or it could be cells in culture even. So just to try and talk about this whole spatio-temporal effect, I've drawn a very bad picture of embryonic brain explant, for example, and two scaffolds. Again, I'm going to talk to you more about the scaffolds in a moment, dipped in blue there and in green. And so the idea is that if you're careful and good with your hands, you should be able to place one of these scaffolds onto the tissue, and that means that the dye or the reagent or the inhibitor or the protein or cytokine, whatever it might be, will seep out over time, but only to that area where the material was placed. All right? And so then the temporal idea, the idea of time, comes that you could put one on before the other, or have them on for a certain time period, like 10 minutes, 20 minutes, or a day, for example, and then you can take it off and analyze what effect that's had. And now, before I get into talking about some of the models that have occurred, I just wanted to introduce you to Professor Anna Williams at the University of Edinburgh. And like with me now, I went up there for a talk, and I brought some of my materials along with me, as I always have them hanging around. I'd gone up to give a talk, and I had a bag of these different cryogels, these scaffolds that I'll talk to you about in a moment, in my pocket, and she came up with this idea. She was the one who first got me thinking about, instead of just developing therapeutics, what about developing tools that others can use in their labs? And she said, hey, can I have one of those sponge things, please? I'd like to use them in my tissue culture. So some of you may be familiar with Anna Williams' really great work, and long before I'd turned up at her to give a talk at Edinburgh, she had already been working on organotypic slice cultures. So I'm sure many of you are very familiar, but if you're not, it's cutting up sections of tissue, as Stuart already mentioned. Spinal cord can be used as well, and then culturing those at the air tissue interface, basically on a filter membrane. Sorry, this floor's very squeaky and I move a lot, sorry. And so she'd been interested in adding a demyelinating agent such as lysophosphatidyl-choline, or LPC for short, and that gave really clear demyelination of the tissue, okay? But one of the problems with this was that if you add something to the media, you get a global effect, i.e. you get an effect on the whole tissue slice. And so the reason she had asked me for one of my sponges, which I'll introduce you to in a moment, as I said, is that her model didn't really recapitulate the patchy nature of MS, for example, or demyelination in MS, where you have some areas of damage or demyelinated tissue next to some normal appearing or relatively healthy tissue. So our aim was, could we try and recapitulate that? So just to introduce you to the scaffolds that we use, I won't go into any of the chemistry there really, but they're called cryogels. And here you can see I'm squeezing it as hard as I can, and it's still remaining intact. So they're extremely soft, like a sponge, yet if you really, really squeeze them, they stay intact. And so the word cryo comes from the idea that you use freezing during the process of making these, but you can make hydrogels from exactly the same materials. This one here is all the same ingredients, everything is the same chemistry-wise, but without the freezing. And as you can see, they are very soft. They're slightly stiffer actually, but they also rupture. So the idea of using these as a tool in your lab potentially means that even if you have a undergrad with very useless hands, you can see you can squeeze it fairly well there with tweezers, as opposed to a hydrogel that obviously, like a very soft jelly, will be difficult to pick up. And that is all borne about by this pore and strut structure they have. So the chemistry is exactly the same, but because of the way you've got all these pores and actually the struts are there make it more robust, you can pick those up. So coming back to Anna's experiment then, she said, well, can I have some of these? And I made them much smaller than the one I showed there as a demonstration. These are about 2 millimeter diameter discs, or 0.5 millimeter diameter disc. And she soaked those in lysophosphatidyl-choline. I say she, but it was her PhD student and postdocs. And they placed it onto the tissue. And the idea was, could we get a small area of pathology in an otherwise healthy slice of tissue? And it's not all just diagrams drawn by me. Here's a example of this, where the cryogel is placed close to this brain tissue. So this would be where, roughly on this picture here, the cryogel would be here. And you get a really clear delineation between the area that's lost its myelin. So you can see there's not much green here for the myelin basic protein. And when you zoom in, it's just debris. So that myelin has been basically removed in this area, but the rest of the brain tissue is still intact. And these spontaneously remyelinate by day 14. So the cryogel is next to the tissue for a short space of time, 16 hours or so, and that gives you that local effect. And just to show their work on spinal cord, again, hopefully you can see the white dotted line up here. So you get this area here that's basically close to the cryogel, that's stripped of all its myelin. And again, you can watch that as that process unfolds of that remyelination. And now you can start to see, well, what are the microglia doing? As you can see, we've stained for IBA1. So what are the microglia doing? How is that remyelination process working? Her group also went on. We made some thinner versions of these to use in vivo. And the setup was kind of similar in a way. Now, these are placed on the outside of the cortex, and she used some that she just added PBS to, and there was no demyelination over the whole time, and others that she soaked in lysophosphatidyl-choline, as shown in this group here, and they got about demyelination. It took a little bit longer, but after about 2 weeks. So there you can see 2 weeks, 3 weeks. This is the control. The cryogels don't have it, and there's no adverse reaction to them, but you do get an area of demyelination close to that. And this was really interesting for her, because this was a way of getting demyelination without the associated inflammation. So they got axonal loss actually occurring here, which meant that the demyelination itself was being involved in neurodegeneration. So this is just an idea of some of the things that we could do with those, but you might have noticed there that we were placing the cryogels next to the slice. But I met Bonnie at the GLIA conference, and she had been working on, she had a poster at the time, but now it's been published incredibly on human post-mortem tissue that they have managed to be able to keep alive. And so here you can see great big huge chunks of tissue being plated in a 6-well plate. So with that, we said, okay, well, we can make some larger cryogels for you if you want, make some larger versions of these sponges. And also, they really wanted to place them on the white matter there. So we had to change the chemistry of our materials to make sure that they wouldn't adhere to the surface of the tissue. And so this is work still in progress, but I want to show you the PBS-soaked cryogel and an LPC cryogel could now be put on the same at the same time. I'm being told time's nearly up. When they're placed on, you can hardly see them there. They're pretty much transparent when they're placed on the slice. But actually, for the LPC one, you could start to see a macroscopic change on the tissue slice there. And when they looked at this here again, using a green marker for myelin, you were getting now an area just of demyelination just underneath where the cryogel was placed, and no problem at all with the PBS-loaded one. So just to fuel your thoughts and maybe think about other things that could be done with this, I'll just run through very quickly some other uses of these materials in other people's labs. I've been working with Vija over in Freiburg in Germany, where we made, I don't know if you can tell what we call these ones, but these donut-shaped ones, it even looks like it's got sprinkles on the top, doesn't it? These cryogels, because again, we 3D print the templates that we make for these, so we can make them in any shapes and sizes. But she really wanted to transplant glioblastoma, so a brain tumor cells, onto tissue slice culture. And the problem with micro-injecting in is that you then get a response to the needle stick injury. So could we transplant these cells and keep them spatially confined, as I've tried to draw as a cutaway view of a donut, with the cells being placed into the lumen of that donut? Could we get spatial confinement of those cells? And actually, yes, was the answer. They would grow in this sort of tight bundle and start to seep into or migrate into the tissue. But quite interestingly, as these brain tumours have hypoxic regions, we were able to show that too. But that's all work ongoing at the moment. One other little video I'd like to show you of this are some line-shaped cryogels, because these really show how robust one of these things is. So that's 0.5 millimetre in diameter and being picked up with tweezers, and yet they stay perfectly fine intact. And so Katie at King's College London, she's done a lot of work with these, and one of the studies she uses human fetal explant tissue, was to see whether she could temporarily inhibit firing, neuron firing, on a slice culture. So she got one of these line-shaped cryogels. She had used Oregon green as a calcium indicator, and she soaked them in tetrodotoxin, or TTX. So this is a sodium channel blocker, and is able to inhibit firing just underneath the area of the cryogel. I've kind of shown that as a darkening of this, but when she measured the intensity, if she placed this TTX-soaked cryogel just on that area, she could get a reduction in the neuron firing. And interestingly, when she took it off with a washout phase, that resumed. And then she could place it back on again, drop the neuron firing again. And so we're able to show that you can place these on tissue, take them off, there's no damage, and that we can manipulate the firing in that slice culture. Only because James was the first to present, and he'd shown some work on growth factor delivery, and he had a graph showing 1 month of delivery. I thought, if you don't mind, I'm just going to pop to one more slide, so I show that our material's working better than his. Sorry, James, I'm only teasing. But this is the same protein as James was interested in delivering, glial cell line-derived neurotrophic factor, GDNF. And we've made these, I just also wanted to show that we've made these all the way down to 20 micrometers. So these little tiny little scaffolds of them, we can get controlled release of GDNF over long periods of time. And if we change how much negative charge we have in those, because we rely on a negative charge in our scaffold and a positive charge on the drug to cause that, we can get faster release if we want. But basically, at the moment, we've gone out to 7 months of controlled release of it. I think there I'd better be quiet, but I did want to say thank you very much for inviting me. Please come up and have a chat if you like afterwards about using these, and thank you for your attention.
I still think we've got 5 minutes.
Okay, great. Okay, yeah, you're right. Sorry. Yeah, thank you.
Yeah, we've got time for a question or two. Yeah, do.
And thanks for a really nice talk, and congratulations on developing such a nice material. So my question is, are there limitations on what you can... Are there limitations on what you can incorporate into your cryogels? And have you systematically studied the time release of various compounds and the kind of distance over which the compounds are released?
Yeah, that's a really good question. And yes is the answer to most of those things. And yes, there are limitations. And yes, we have looked at things. So to deliver anything for a short period of time, it's very easy. Okay, so if you want to deliver these over a period of day or two, you can literally soak them in. There doesn't have to be any kind of interaction. We've used all sorts of different drugs, and that can work. If you want the long controlled release, as I kind of alluded to with my shaky hands, if you have a negatively charged scaffold and a positively charged drug, then they will electrostatically attract, and that will slow the release over time. And so we're actually looking at repurposing drugs for brain tumors and have done a big wide range of studies looking at loading and release. And it looks like the limitations are that we can only work with charged drugs if we want sustained release, and that small molecules aren't as easy to control the release of as proteins. With that really long release I showed of GDNF there, we can replicate that with PDGF, with BDNF and other proteins, but getting long release of small molecules, we're in the order of months, nearly 2 months, rather than half a year and onwards. So it is harder to release the small molecules over a long period of time.
Can I ask another quick one? And the materials themselves, the cryogels, I couldn't get a sense of how the microglia see them, and do these degrade over time if you put them in vivo?
Okay, really, really good questions. Yeah, so I haven't shown any of the work on Parkinson's and using these as a therapeutic. To be short on one of the answers, they're not degradable. We are looking at making biodegradable ones, but I quite like the idea that once you've implanted these, there's no change in that microenvironment. They're getting the one-off shot of being put in there, but they're not, there's not that constant degradation and that constant remodeling of that environment. As for having a look at microglia and what they do, when we've implanted these into the healthy rat striatum, we see an influx of microglia as we look with IBA1 staining, and we get the sort of general sort of hue of astrocytes around them. But that is no different to a needle stick alone injury. So we compared that with a needle stick, with just an injection of PBS or with the carriers, and that basically shows us that they're pretty inert.
Probably got time for one more. Yeah, Stu.
Yes, you mentioned cells being recruited to them, but are there actually the cells invade into the material? Do the cells climb into it, or?
So yeah, in the ones that we injected for the Parkinson's work were these 20 micrometer ones, and you can see that there's absolutely no room for a cell there in those pores. But we have also been making these ones with large pores. Now these are about 200, 300 micrometers in size for cell delivery. And Jerome in San Diego has been adding in what he calls induced neurons, so they're directly reprogrammed neurons with astrocytes into them, and you get these lovely processes out. But again, this is a slightly different chemistry. All of this sounds like it's easy with one type of chemistry, but we've actually mixed and matched. So we make these positively charged, and the cells can then attach to those and grow throughout. So, but we haven't put those in vivo yet.
That's great, thank you.
Cool. I think we'll
Thank you very much.
Interesting time, thank you. So yeah, that just remains for me to close the session. I realise we are quite short on time, but one thing we wanted to do is enhance the visibility of neural tissue engineering, and potentially what we're looking to do going forward is creating a more national and international network within which we can share ideas. So groups like this are going to be really vital for that. And I guess watch this space for what we'll do in the future. But if these kind of things interest you, please come and talk to us or reach out to us, and we'll see what we can do with that in the future. There's plenty to do, as you can see from all the fantastic work. Lots of things that can still be advanced, including in the modelling aspect. So modelling neurological disease, developing those representative models using tissue engineering approaches. So I'm going to finish there, but I'll just finish on thanking all of the speakers. They were really great talks and really engaging. And thanks everyone for being here as well. Thank you.